Welcome to this webinar. In this case study, we want to discuss how to translate a laboratory single-cell RNA-Seq workflow into a cloud data platform for analysis of single-cell RNA-Seq data. My name is Jonathan. I'm the founder of EVOBYTE Digital Biology. EVOBYTE enables research teams from biopharmaceutical and life science sector to manage and analyze their data. And before we go into the actual webinar topic, I would like to give a short perspective on what EVOBYTE offers its clients. So EVOBYTE designs and implements customized data platforms that are built around research processes, around laboratory processes, with the aim of capturing all information and outputs that are produced by this research process or related to the process into a single repository. And there are typically two types of input data that are modeled into the process data layer. And this is instrument raw data, so measurements that are produced. And on the other hand, documentation, annotation data, and metadata. And the key step is to define a data structure that is able to link both the annotations and the raw data, and to save those structured data into a database or data repository so that the connection between those data types are preserved. And this can include additional processing steps. So for instance, for next generation sequencing workflow, as we will see today, the process data layer also encompasses automatic data analysis and processing steps so that there's a minimum of hands-on time required. And once we have such a structured data repository in place, EVOBYTE is also offering different types of downstream analytics that can be implemented on top of this. This can, for instance, be visualization tools or data apps. This can be tools to create automated reports. This can be integration into data warehouses, for instance, or data export functionalities, or of course, additional machine learning, data science, or bioinformatics to further dive into the data that we've collected beforehand. And again, the key is to produce a single structured storage so that clients can rely on having all important information that are produced there, all the value that is being produced in their research processes into a single, secure, and lasting data structure. So what's going to expect us today? We will have a look at a single-cell RNA-Seq laboratory process and try to get an idea of how we can model such a process and the data that are produced there into a process data layer to capture all important information. Then we will have a look at how a user would basically interface with such a process data layer, and we are going to have a little live demo to actually look at such an application and how to add information to the datasets that are being produced here. And finally, we will have a technical deep dive into how to set up such a infrastructure on the AWS cloud. We want to have a look at a research process, and in this case, we will look at a single-cell RNA-Seq workflow. And we assume that we have a startup company that wants to perform some routine single-cell RNA-Seq analysis of blood samples that are collected from patients in order to measure gene expression in each single cell. So for this, the first step would basically be to prepare a cell suspension from the PBMCs. And this may include changing buffers, for instance, and counting the cells as input for the single-cell RNA-Seq experiment. Then the next step, we would perform the actual single-cell RNA-Seq experiment that would include loading the cells to, for instance, a 10x controller in which the individual cells are encapsulated into droplets and then lysed, and the mRNA from each of the cells is captured onto a bead. And then these beads can be collected and processed into next generation sequencing libraries. And those libraries can then be sequenced on an Illumina machine, for instance. And after the sequencing, we assume that we will also directly run the demultiplexing step on the sequencer or on a computer that is attached to the sequencer, so that we basically end up for each of the samples that we analyzed with a set of FASTQ files, and these FASTQ files contain our read information from the sequencers. So this is basically our instrument raw data in this case. And on the other hand, as I said in the beginning, it's very important to link the appropriate metadata so that we basically capture the full experiment on this platform into our data repository. And in this case, we decide that we want to go for a very simplistic metadata set. We will collect the date of the experiment, an experiment ID, and some additional information, for instance, a protocol ID or an SOP, and a patient ID. And this we want to basically later link with the actual single cell data set and store as metadata in our platform. So this is basically the basic data inputs. And second, there are a few additional steps we would like to implement to automate the data analysis. And this is first, we want to run an automated analysis of the raw data. So these FASTQ files need to be processed and analyzed to actually get to the gene expression measurements for each cell. And this step we basically want to automate. And then second, we want to add another automation step in which we want to integrate the current data set that we upload with all of the previous data sets so that we can do a clustering of basically the whole history of data that we collected for this type of workflow. So this is basically the second additional functionality we want to implement into our process data layer, and how this is done we will have a look at now. So this is now an overview of a concept for the process data layer that incorporates all the requirements that we defined in the previous step when we looked at the actual laboratory process. And as we said, there are two types of inputs. So we have basically experimental metadata, which we need to ingest. For this, we use a web application. And second, we have basically the raw FASTQ files, which we need to upload into the process data layer that come from the next generation sequencing experiment. Let's have a look at the metadata first. So the idea here is that we basically provide a web app in which we list all the single cell RNA-Seq data sets, and then a user can navigate to this web app through the browser and simply add the metadata there. So this makes it very easy because it's very flexible, and there's also additional functionalities which can be included into the web application. And in this case, we decided that we would like to have the functionality to basically annotate and browse the RNA-Seq data sets. So that refers mostly to the metadata, but then we would like to have two additional functionalities. And one is basically we want to have a view in which we can check the QC metrics for each of the data sets so that we can right away ensure that the quality of these data sets is decent. And second, we would like to have a basic data exploration utility in which we can have a look how the integrated data sets look like and do some first set of comparisons. And then on the side of the raw data, as I said, there are three steps or three functionalities we would like to implement. First, we want to basically perform a very basic QC, in which we check that the FASTQ files actually have the right type of format, that they are not corrupted or anything. So this would basically be the registration stage. Then we have basically the raw data processing, and this includes performing the alignments and the gene quantification, so the actual analysis of the single-cell RNA-Seq dataset that leaves us with the gene counts for each of the cells that we sequenced. And finally, we would like to perform the integration step in which we combine multiple datasets. And the important thing here is now that each of these datasets should be linked to the metadata in our data app, and they should also be, of course, stored and structured in a relational database so that the information is preserved and can be securely stored on a proper database server. Let's have a look at the bioinformatics steps that we want to implement for this pipeline. And as we discussed, the first step is a registration of the FASTQ files once we uploaded them into an input bucket. And there are a few things that need to be checked here just to make sure that we don't execute the downstream processing steps on files with errors or files that don't have the right format. So we're checking, of course, if the FASTQ files are paired. So if we find a read 1 or read 2 that we always expect for single-cell RNA-Seq sequencing. And then we also check if the file format is valid. So if it's really a FASTQ file or not accidentally another file. And if that is true, the FASTQ files are registered and stored in our database. And then the next step is being triggered. And this next step is basically the raw data processing. So this executes, in essence, the single-cell RNA-Seq pipeline. This can, for instance, be the Cell Ranger software from 10x Genomics in case we use this as a platform, or any kind of custom script, for instance, when you want to run a drop-seq experiment. But what is happening there, in essence, is that the reads are aligned to the genome. Then for each read or for each set of paired reads, this read set is also referenced to the cell barcode that is sequenced, so that later for each cell that were sequenced, all the reads can be collected and the genes can be quantified for each of those cells. And in the end, we end up with a digital gene expression matrix. And this basically contains, for each cell that we sequence, or each cell barcode here in the rows, the gene counts in the columns. And again, we store these digital gene expression matrices together with additional QC files that come from the single-cell pipelines in our database. And if this step has been successful, we automatically trigger the third step, and this is basically the dataset integration. And during this integration step, we first load all the digital gene expression matrices that we collected throughout our workflow. So not only of the current experiment, but also of the previous ones. We then filter and normalize these data sets. We remove cell doublets that can arise during the experiment, and then on the full data set, and this is important, we calculate a neighborhood graph and perform a clustering of the full integrated data set. And additionally, in this case, we also calculate a dimensionality reduction, so a UMAP embedding that we can later use to visualize these data sets and all the clusters that were found for this integrated data set. And again, this integrated data set is also stored in our database and basically marked as the latest version, since there's always, for the whole workflow, one up-to-date, latest integrated single-cell data set that we can use for downstream analysis. Finally, we want to have a perspective from the user, how to interface with the process data layer. As we said, there are two types of input data that need to be ingested, and first there's basically the raw data that come from the next generation sequencing. And then we have the experimental metadata, which we need to annotate each data set with. And first, regarding the raw data, so those would be the FASTQ files that are produced after demultiplexing the actual sequencing runs. And I mean, typically these FASTQ files would reside either on the sequencer itself, if you have newer sequencer versions, or possibly in most cases on a file server that is somehow attached to the sequencer itself. And what we would need to do now is basically upload those FASTQ files into an input bucket. So this is basically an AWS S3 bucket, so some sort of like a folder in the cloud in which we upload the FASTQ files. And these upload would then automatically trigger the execution of the FASTQ registration steps and the downstream processing of the FASTQ files. And such a process can, of course, also be automated, so it's very easy to set up a trigger that is automatically uploading new sequencing files after the FASTQ files were created into the S3 bucket. And otherwise, the user would basically need to manually perform this upload into the input bucket after each sequencing run. And second, there's the web application in which we upload the metadata. So here we basically tag each data set with the metadata that were collected for this experiment, and how this looks in practice we will see now. We have now logged into our data web application and moved to the annotation page here. And the annotation page provides a list of all single-cell RNA-Seq datasets that have been processed. Each of these datasets gets a unique identifier by the process data layer and is also connected to the FASTQ file of origin that was basically input into the single-cell RNA-Seq pipeline for analysis. And additionally, we see all columns which we wanted to define to annotate metadata for each of the datasets that we have processed here. This is a name column where we can specify a more human-friendly alias instead of these bulky IDs that are also later on shown on the plots that we can produce. We have a description column here where we can add additional descriptions for each of the data sets. And then we have a date column, which refers to the processing date, a replicate column, a protocol ID column to store information, how the cells were processed, for instance, or an SOP version in this case, and an experiment ID column where we can specify the ID of the laboratory experiment. We can very easily update these entries here. So let's say this is not experiment A4, but experiment A6. Then we can just simply change it here, press the save button, and then these changes are also stored in our database while still being linked to the single-cell RNA-Seq dataset. And we can very easily add other columns here. So initially we wanted to add a patient ID column. We can simply add this and it will appear here in our table. And then we can basically add a patient ID. So this is patient 1, 2, 3, and 4 that we have analyzed. We save this. And then also this new column is stored in our database. Similarly, we can perform annotations on the single-cell RNA-Seq clusters that were found in the integrated dataset. But for the moment, we will move to the metrics section and get an overview of the QC statistics for each of the datasets that we have analyzed so far. So here we see these violin plots that show the number of RNA molecules that were identified for each cell in each of the datasets here, or the number of genes that were detected. These are some fairly standard readouts for a single-cell RNA-Seq analysis. and also to compare the quality between the data sets. We can also move to this QC section here. These are further metrics that are QC related, for instance, the fraction of mitochondrial reads in each cell, which is typically an indicator of the cell quality, or of cells were basically of low viability at the point of processing. These types of QC metrics can be looked at here in this section. And finally, we can move to the Explorer section. This is a interface for exploratory data analysis. Here it's fairly simple. So it's a UMAP representation of the clustered data sets. We have implemented a step in the process data layer, which is performing an automated integration of all data sets that were part of the analysis. This means that those 4 datasets here were automatically integrated and clustered into one single large dataset. This saves us a lot of hands-on time and allows us now to look at the data from the whole workflow in one plot. The colors in this case are the individual data sets here, and we can see that most of the larger clusters are comprised of cells from all of the data sets except this one here, so that would be interesting to look at now. And we can also select and change the grouping here towards the Leiden clusters. So this is basically the single cell clusters that were identified by the Leiden algorithm, and that are then in some way representing our cell types or our cell states. And these are the typically a first point for a downstream analysis that we can perform here. And with this, I would like to summarize the user interface. So there were 3 main functionalities which we implemented here, which is an annotation, a metrics, and an exploratory data analysis section. And each of these help us to streamline our analysis, to automate our analysis, and to capture all relevant metadata in a very simple and accessible manner. After this live demo, I would like to summarize the first part of this webinar. So we've seen how we can go from a laboratory process for a single-cell RNA-Seq workflow on blood cells to a concept for a process data layer that is able to capture and store all relevant information and data that are produced by the research process. We've seen how to design consecutive data analysis steps that are automatically triggered once new raw data arrive. So in this case, we first check that the FASTQ files are actually valid and have the right format. Then we trigger the automated analysis of the single-cell RNA-Seq raw data using a standard pipeline like the 10x Cell Ranger or any other pipeline that one would like to implement here. And third, we add a dataset integration step, which is automatically triggered, that takes the current and all the previous datasets and performs a combined filtering and clustering, so that in the end there's a single integrated dataset available for downstream analysis. And then we have seen how to interface with the single-cell RNA-Seq data using a web application, which the user can add the metadata and tag each dataset and perform some first QC analysis and exploratory analysis on the single-cell data. If there are comments or questions, please leave them here or reach out via LinkedIn or info@evobyte.com. And we will continue now to look at the technical implementation of the process data layer in the AWS cloud. So let's have a look at the technical backbone of the process data layer and the data platform for our webinar here. So in general, data platforms developed by EVOBYTE are tailored cloud-native applications, so they would typically be hosted in a client's cloud provider account, and then really customize to a client's needs and to the research process that should be modeled and which data should be stored within such a data platform. In this case, this application here is implemented as a sort of highly containerized architecture that can also be scaled very flexibly, and that is also leveraging a lot of the security features that cloud computing environments can offer. So here, basically, for the process data layer, the backend is a Django web framework. So basically a Pythonic backend that is connected to a PostgreSQL database. And this database is basically hosted as using the AWS RDS service. So this is basically a managed service by AWS providing the database, and that allows us to very flexibly scale those databases and manage backups, and it also increases the security of the database application. Then we have the user interface, and this user interface is implemented as a Streamlit data application behind a reverse proxy to enable encryption. And then we have a number of bioinformatics steps that we've seen before, which are implemented as Docker containers, and each Docker container takes care of an individual step. And those are actually only triggered when there's new data available. So what this means in practice is that once we upload a new data set that comes from the sequencer, this will basically trigger a first container task that is running on the AWS ECS service using a Lambda function. And once this task is triggered, it will basically execute the first Docker container, which takes care of the FASTQ registration, and then communicates with the backend to save and store the new FASTQ files within or in the database. And once this is complete, this would basically then trigger the next container, and this next container would contain the raw data processing. And once this is complete, it would automatically trigger the next container. So this kind of on-demand execution of tasks saves us quite some resources. So we only have to pay for what is used here in case we actually have new data. This is quite economic, and it's also quite scalable. And this is the second big advantage. So looking, for instance, at the raw data processing step, this is kind of resource intensive, needs a lot of CPUs, needs a lot of memory to run the alignment algorithms and so on. So we can explicitly for this task configure more resources as we would, for instance, need for our FASTQ registration step. And the same is also true for the dataset integration. So while it may seem a bit granular to set up these 3 different containers, in practice, it's actually very flexible and dynamic to set up these types of architectures, because you can also update these containers individually, or we could add another container that has a different type of functionality, and then we can really leverage the power of the cloud to perform these on-demand batch execution, so we don't have to pay for any resources here that we don't need. The only resources that are permanently running are basically the web applications and the backend. Next, we want to have a look at how to deploy our application actually in our AWS account. So for this, we first define a VPC, so a virtual private cloud, which is some sort of dedicated network for our application within our actual AWS cloud account. And within this VPC, we can define a public and a private subnet to host our actual application components in. So this private subnet has the advantage that it's not reachable and cannot communicate to the internet, which is of course increasing our security for the components that we host within the private subnet. In our case, this is the managed database service as well as the Django backend. And also the bioinformatic pipeline steps, so the containers that process our RNA-Seq datasets, they are also executed within this private subnet. So none of this can communicate to the internet, and so also the internet cannot communicate with these applications, which makes it quite secure. And then our user interface is hosted in a public subnet with internet access, and this would basically be the Streamlit app and the Nginx reverse proxy, which can be reached from the outside, and then the users can log in to these applications with their user accounts and basically access the datasets they have the permissions for. One could even increase the security here and also place the user interface in a private subnet. Then it would only be reachable via a VPN network, which could also be configured. And then the file server, which hosts our FASTQ files, so the raw data from the sequencing, they would basically perform an upload into a dedicated S3 bucket. And also these buckets can be configured in a way that they are only accessible from a dedicated host, so only from one server, that we can restrict the permissions to, so that also here we are able to really encapsulate our applications. And finally, this whole stack is implemented as a CloudFormation template, so following the infrastructure as code principles, meaning that the exact same application can be very easily deployed in different accounts or to run production and test parallel workloads, which is another great advantage of bringing this in as a fully cloud native application. And with this, I would like to summarize the second part, where we had a look at the technical implementation of the data platform. We've seen a cloud-native application that is able to really leverage the flexible design patterns and the scalability that can be leveraged using cloud computing. For instance, we can define steps that have very specific requirements in terms of CPU and memory, and only trigger those steps and also only pay for those steps when we actually really need them, so that they're not idling. We've seen how to automate a data pipeline with several consecutive steps using Lambda functions and containers that are basically chained based on the output of the previous container or the success state. And each of these containers then individually communicates to our backend and ensures that the data that are produced by this container are also stored in our database and made available to the users in the end. And finally, we had a look at how to deploy these applications in a cloud account. We've seen how to set up private and public subnets to maximize the security of our application and to encapsulate data in a way that they are highly secure, stored and backed up in the AWS account. So we reached the end of this webinar, and I hope you enjoyed it and you could take some ideas and inspiration how to set up a data platform for your research projects. If you have questions or comments, please leave them below the video or reach out via info@evobyte.com or via our LinkedIn accounts. Please visit evobyte.com if you want to see some more webinars and some more information on related topics. You can also get an idea of the services that are offered by EVOBYTE on our website. And if you have a project inquiry or an idea or something related to setting up a process data layer for your research process, please feel free to reach out. And with this, I would like to thank you for your attention and wish you a great day.